Okay, so you're trying to like learn new things quickly, right?
Yeah.
No one has time to read like, endless textbooks and stuff. So we try to get you those core insights on like a bunch of different topics,
Mm.
and today's no exception.
Yeah.
Today we're going to do a deep dive on a plant biotechnology technique
Hmm.
that's been making some noise,
Hmm.
It has a lot of potential for making valuable compounds really efficiently.
Right.
And like the source material that we're looking at today is called
Right.
Production of Secondary Metabolites in Plants through Hairy Root Culture. So
Yeah.
basically, we're talking about
Okay.
harnessing nature's chemistry in a new way.
Exactly. Yeah. And I think what's so fascinating here is this technique we're talking about, hairy root culture, is almost like
Yeah.
a clever shortcut to getting these compounds that everybody wants, right?
Yeah.
So our mission is to break down how it works and really why it's so significant.
Okay. So a hairy root culture, I mean, it definitely paints a picture, right? Like,
Yeah.
what are the basic ideas, the principles at play here?
Sure. So at its core, it's a plant biotechnology method. And what makes it so unique is it uses plant roots that have been transformed by a naturally occurring bacterium
Okay.
called Agrobacterium rhizogenes.
Agrobacterium rhizogenes. That sounds like something straight out of a,
Right.
science lab.
Yeah, it is pretty ingenious. This bacterium has this really cool ability to transfer some of its own genetic material
Uh-huh.
into the plant's root cells. And this transfer involves something called the rhiplasmid.
Okay.
And the end result of this natural genetic modification is the roots exhibit really fast growth, and this is what we're interested in, become real powerhouses for producing secondary metabolites.
Okay, so secondary metabolites, we've been saying this term, like,
Right.
what are they exactly and why should we care?
Good question. So think of them as specialized chemical compounds that plants produce naturally for things like defense mechanisms or communication. And what's really interesting is that a lot of these compounds are valuable to humans for use in, pharmaceuticals, flavorings,
Mm.
fragrances, you name it. Yeah.
For example, paclitaxel, which is a really important anti-cancer drug,
Right.
is one compound that researchers are really excited about potentially producing
Yeah.
more efficiently through these hairy root cultures.
Wow.
And it's important to remember that all of this, growing these roots in a very controlled lab environment, falls under the umbrella of tissue culture.
Okay, so it's like we're taking advantage of something the plant can already do, but we're doing it in a way more controlled and potentially more efficient way.
Yeah.
The source material also talks about how we need to establish and maintain these cultures.
Right.
What does that even mean?
Yeah. So while our source didn't really dive deep into the specifics, it's important to understand that this isn't just a free-for-all kind of process.
Right.
Establishing a culture involves very carefully introducing the Agrobacterium rhizogenes to a specific plant
Okay.
under very controlled conditions.
Yeah.
And once the roots have been transformed, they need to be isolated and grown in this nutrient-rich medium in the lab.
Oh, wow. With constant attention to the environment to make sure they keep growing and producing those metabolites.
Interesting. So we got these special roots now thriving in the lab.
Exactly.
Let's talk about why this is getting so much attention.
Sure.
What are the advantages and the implications here?
So the main advantage, as our source really emphasizes, is this efficient secondary metabolite production.
Okay.
And these transformed roots, they often grow much faster than normal, and they produce higher and more consistent levels of these valuable compounds
Wow.
compared to traditional farming methods.
Yeah.
And if we zoom out a bit, you can see how this could lead to a more reliable and sustainable supply of these important substances,
Right.
potentially bypassing issues like seasonal changes or
Yeah.
geographical limitations.
So it really is about getting access to these valuable things quickly.
Absolutely. Yeah, this efficiency really opens up a lot of doors for different applications.
Yeah.
Particularly in the pharmaceutical industry where consistency of quality and supply is really crucial. Imagine if we could produce life-saving drugs in a much more reliable way.
Okay, so to summarize this deep dive we've been on,
Yeah.
hairy root culture, this plant biotechnology technique
Yeah.
that basically uses this natural gene transferability of Agrobacterium rhizogenes
Right.
to create these fast-growing roots that are really good at producing those valuable secondary metabolites.
Yeah,
And it all happens in a controlled lab setting.
exactly. That's a great summary, and I think it really highlights our growing understanding of plant biology and how we can use it for real-world benefits.
And it makes you wonder, given like the sheer number of compounds plants can naturally synthesize,
Right.
what other, ingenious methods might be just around the corner to unlock even more potential.
Yeah,
I feel like we're really just getting started here.
absolutely.
It's so exciting.
It is.
Thanks for listening, everybody.